Okay.
We're recording in stereo, so if one fails, I think we'll still be okay. Do we have any questions on what we covered last time? No. Good. We're going to touch on the RNA basis of toxicity today. It's going to be my extra point because I'm an idiot and I don't really know what you all know coming in here. I just want to make sure we're all on the same page as far as what goes on in gene expression and why double-stranded RNA should just pop up immediately as saying, okay, that's abnormal. So we'll get that. The main point today is, of course, ALS. So here's Stephen Hawking when he was alive and much younger, before ALS left him wheelchair bound. Is this dark? It seems very dark. Okay. So we'll go over ALS, what it looks like, what causes it, and then after we wrap that up, we're going to talk about microRNAs and RNA toxicity. It relates to ALS. It relates to Huntington's. It relates to any polyglutamine disorder. There's a bunch of them. You guys have some questions? We'll start at the top. Why do we see a higher incidence of ALS in military personnel and soccer players? I like maybe it's because they tend to smoke or they come in contact with pesticides. Probably it's just repeated head injuries. That's demyelination. Why does it spare the posterior columns, and what does that mean? Two excellent questions. You always want to know what something means. So what that means is that the sensory pathways in our spinal cord are spared. Since your tactile sensation runs up the posterior columns, the fact that you're not seeing demyelination or reactive gliosis in your sensory pathways, but only in the motor pathways, tells you that ALS primarily affects motor function rather than sensory. So it's a motor neuron disorder, not just a general neurodegenerative disorder. It's specific for motor neurons. Why? Well, it probably has to do with the difference in gene expression profiles that motor and sensory neurons display. These two different types of neurons have to express different genes. Otherwise, they wouldn't have different functions. And really, ALS all comes down to an impairment in proper gene expression at the RNA, protein, or trafficking levels. And so since sensory neurons don't express the same types of genes, they're not going to be susceptible to the same types of mutations. So while every cell in our body has the same DNA in it, they all make different proteins. They all make different RNAs. And so depending on which RNAs you're making, which proteins you're making, that's going to determine which mutations will affect you. It's a long-winded way of saying I don't know. Bulbar onset. Anyone know what that is? Something to do with here, exactly. Brainstem. Yeah, so you see your symptoms primarily arising in structures controlled by the brainstem. So you'll have difficulty speaking, swallowing, moving your face, things like that. Let's see. Does that mean that multiple repeats hit the brainstem for anything else? Really what it says is that your brainstem carries out vital functions. We've heard this before, and it's still true. So your brainstem is going to carry out respiration, swallowing, speaking. So if you have your neurodegeneration starting in the brainstem, that means that your brainstem is going to be more profoundly affected by the disease than if it were to start, let's say, in the limbs. And so you see a year or two difference in their lifespan, depending on whether you have bulbar onset or more lateral limb onset. Yeah?
Do you know what would cause bulbar onset versus like onset in a different area or peripherally or something like that?
No. No, I don't have a specific answer to that. Yeah. for whatever reason, those motor neurons are more susceptible. There's a variety of mutations. I don't know if any are more associated with bulbar onset as opposed to lateral onset. It's a great question. I just am not smart enough to answer it. Sorry. So by scavenging free radicals, you mean SOD1 is getting rid of them? Absolutely, that's what that means. The function of superoxide dismutase is to neutralize those free radicals that we always make. So the assumption is then that, and if you were to only read the book, that whenever we have mutations in SOD1, that it doesn't do its job anymore, and so it's a buildup of free radicals. And that sounds wonderful, but it's totally wrong. That's not what's going on. It has nothing to do with the loss of function. The ultimate loss of function is a knockout. When you knock out SOD1, no more SOD1 in that animal, yet you don't see motor neuron degeneration. When you completely get rid of the enzyme, as opposed to having a 10% reduction in its efficiency, you don't see motor neuron degeneration. So it's a gain of function. What do you mean by gain of function? Exactly what it sounds like. Don't overthink it. That mutation causes the gene to gain a function, not lose its normal function. So it's gaining some toxic function rather than losing a vital function like free radical scavenging. It can still do that. It's not quite as efficient, but many mutations have no impact on the catalytic side. SOD1 still works. SOD1 still neutralizes free radicals. But it does something else too. And we'll get into that. So it's all about the loss of motor neurons in ALS. We've seen, well, we've certainly seen the basal ganglia at this point, PD, HD. Both of these diseases affect the basal ganglia. Basal ganglia are very important in regulating motor function. That's the filter. Should I do this or not? I don't know. Let me ask my basal ganglia. Direct pathway will say go, indirect pathway will say. That's the oversimplification. We're fine with that until next semester. The cerebellum is also really important. That allows our movements to be fluid, automatic. I don't have to think about this. I'm walking around. I'm pretty proud of myself. I can walk and talk at the same time. It's because I have a functioning cerebellum. And the only reason I have any ability to move is because I have upper and lower motor neurons. My lower motor neurons could be in the brainstem. This would be in our cortico-bulbar tract. There's also the cortico-spinal tract that heads down to the lower motor neurons in the spinal cord to control areas outside the face. So the brainstem is going to control muscles of the head and neck for the most part. And then in your spinal cord, that's where you're going to get a little bit of neck as well, and then the rest of the body. Upper, it's up high in the cortex. Here's my upper motor neurons. Lower motor neurons are lower than upper motor neurons. Without your motor neurons, no movement. But without those supporting characters, improper movement. So we've seen what happens with basal ganglia damage. If you have damage to your cerebellum, you see these gait abnormalities. But today it's all about the motor neurons, because that is what we lose in ALS. This disease specifically targets upper and lower motor neurons. You'll get a different mix from patient to patient. And you'll see the death of motor neurons. Age is of course highly relevant to disease onset, just like every other neurodegenerative disorder. The older you are, the more likely you are to have enough neurodegeneration to see symptoms. Almost all cases are going to come on after 40 years of age. Not all. We'll see a young man today. But in most cases, it's going to be later in life. This tends to affect males, more so than females, and whites as well. So we see higher prevalence in white males. This is usually not inherited. The majority of cases are sporadic, no familial history. The risk factors that you might be able to control would be exposure to pesticides. This is still a bad thing. In this case, there's probably some underlying point mutations that make their motor neurons more sensitive to death. And so when you perturb mitochondrial function with pesticides, well, then those somewhat stressed motor neurons die off. So pesticides don't only affect dopamine neurons, they just tend to die Smoking is another risk factor. The earlier one starts smoking, the higher their risk of developing ALS compared to never smokers. The criteria for ALS would be really this rapid loss of musculature and motor function. The onset can start in the body or it can start in the face. So we're going to see the weakness and muscle wasting would be in the limbs. It could also start as just difficulty with speech, swallowing, and then spread to the limbs. This would be our bulbar or pseudo-bulbar palsy. In bulbar palsy, it's the actual death of the lower motor neurons in the brainstem. In pseudo-bulbar palsy, it's the death of the upper motor neurons that innervate the brainstem. So in both cases, you're affecting control of the face. With bulbar palsy, it's lower motor neuron death. With pseudo-bulbar palsy, it's upper motor neuron death. But in both instances, you see the initial symptoms affecting the face. And this bulbar onset, whether it's pseudo or actually bulbar palsy, is predictive of poor prognosis. That's because your brainstem carries out vital functions. If you have difficulty breathing, that's a lot more serious than difficulty clasping your hand. As the disease progresses, you're going to see muscle stiffness. You'll see wasting of muscles, of course. You'll see these fasciculations occurring and widespread weakness. It's going to affect at least 3 of the 4 limbs. You'll see different degrees of weakness in the lung, in the limbs versus diaphragm. You'll see a lot more of a weakness and difficulty breathing with the bulbar onset. But eventually respiratory distress occurs across the board. Sensory function is preserved. So regardless of whether you're seeing degeneration of your cranial nerves, your cortical bulbar or cortical spinal tracts, or your lower motor neurons in the spinal cord, sensory function is still there. And these red names are just to give you the different types. So both pseudo-bulbar and bulbar palsy are affecting the brainstem first. It'll spread. The primary lateral sclerosis or this progressive spinal muscular atrophy, these start in the limbs and eventually spread up as well. This is a progressive neurodegenerative disorder. And we'll see how rapidly this progresses.
It's a bigger question. Yeah. The first question is, how are you doing? Fine. And the second question is, how are you really doing? And that's a much longer conversation.
It's weird because it seems like it's like all different person.
It was only 6 months ago.
Yeah, it wasn't that long ago.
When you see yourself 6 months ago talking about ALS and about your friends, about how you're dealing with it, what are you thinking when you see that now?
I think how lucky that guy was at the time.
So in 6 months, pretty rapid progression now. It's a lot more difficult to understand it. You can see the wasting away that's occurring. Now you can distinguish whether it's the loss of upper or lower motor neurons depending on the type of symptoms that you see. So we have degeneration of upper motor neurons. Both of these are going to cause weakness or paralysis. But they affect the muscles differently. Now immediately after the loss of upper motor neurons, you may see just flaccid paralysis, just complete weakness. This is more common in something like a spinal cord injury, though, where you get a severing of a large bundle of motor neurons. If that occurs, afterward you will see something a little different. Rather than a flaccid muscle, you'll actually see an increase in muscle tone, so it becomes spastic. You'll also see hyperreflexia. So reflexes become a lot more pronounced. You may also see abnormal reflexes, depending on which upper motor neurons you're lost. One of the purposes of the corticospinal tract or corticobulbar tract is to inhibit reflexes. If you want to carry out a voluntary movement, you have to inhibit the reflexes that prevent involuntary movements. So if your leg changes, so if one of your muscles changes length without you doing it, hopefully we remember the stretch reflex is going to cause contraction. So part of carrying out any movement is to silence those reflexes that resist changes in muscle length. So when you lose your corticospinal tract, those reflexes can just take over. And they become far more robust. They're no longer controlled properly. You'll certainly see weakness because your ability to control your muscles is impaired. You've lost motor neurons, in this case upper motor neurons. You might see muscle atrophy. This is not from denervation, but instead just from disuse. They're just not being used enough.
Then we're going to check his L4 on the
You'll
left
see
side.
the differences in reflexes here.
Quite hyperreflexive. S1. Okay, now we'll be careful on this side. Okay, so L4. Okay, and look, we have some clonus in the leg. We can stop it by pushing on the Golgi tendon organ just above the knee. That's a significant sign of some sort of myelopathy going on or upper motor neuron lesion. So what we'll do here is we'll gently dorsiflex him, we'll check his S1, and it actually activates his L4. He's so hyperreflexive. He's unable to dorsiflex the big toe. He's unable to dorsiflex the foot against the tone there. And his inversion and eversion are about 2 plus over 5. Another very interesting finding, and we'll zoom in on this one, is that he has a very positive Hoffman's test. So I'd like you to relax your hand. Okay? And what you do here is you hold the middle finger, you flick it, and observe the thumb and the index finger coming together.
So sometimes you'll see these abnormal reflexes emerge. Like a jaw jerk reflex, you shouldn't have that. But if you have pseudobulbar palsy, this reflex will emerge. Normally it's silenced, but the loss of those upper motor neurons unsilences it. Lower motor neurons, on the other hand, they're going to cause muscle weakness as well. They're going to cause the wasting of the muscle far more aggressively than upper motor neuron loss. You're going to see a decrease in muscle tone. We no longer have motor neurons innervating that muscle. And you're going to see the loss of reflexes. You can't have a reflex arc if you don't have a motor neuron coming back to that muscle to cause contraction. So looking at reflexes tells you a lot about the location of damage. Hopefully you remember that when you go to test them. You'll also see these little flickerings, which maybe we can see here. It's a bit dark. You see his muscle there, just contracting on its own. Involuntary contractions. You see this a lot in motor neuron disease. This can also occur without the overt loss of motor neurons, so don't worry if you get that little eyelid twitch. It's okay. If you cut down on the caffeine, your motor neurons are just a little more excitable. Should be. It's all right. So the differences in reflexes cannot be distinguished upper versus lower motor neurons, but both are going to occur in ALS. And that's going to lead to the wasting of muscles. It's the name amyotrophic. Here we can see a cross-section through a muscle. Healthy ALS. Notice these small degenerating muscle fibers. They're not nice and uniform. And that's caused by the loss of motor neurons. Here we can see the loss of motor neurons. We're looking at it, yeah.
Is there any pattern to the muscle wasting, or is it random?
Do you mean in types of fibers?
Or yeah, that picture that you showed prior. Is there any pattern to where they muscle waste, or is it just kind of widespread?
Any fiber that's no longer innervated by a motor neuron. Yeah, those are going to waste away. So there's a back and forth talk between motor neurons and muscles. They keep each other alive. And so you lose either one, the other one's going to waste away.
Okay.
So here we can see the loss of motor neurons here in the ventral or dorsal roots. They're flipped for whatever reason. So your dorsal root is down here. Here's the cross section. Here's the cross section through the ventral root. And all those little circles there, those are axons. look over here, here's an ALS model. Fewer axons. Fewer axons because you have fewer motor neurons. In the dorsal root, things look just fine though. We're not losing sensory neurons. This is showing us that there's specificity. ALS is a disease that targets motor neurons, both upper and lower, but not sensory neurons. When you look in the neurons themselves, you'll see protein aggregates. Here it is, we call them inclusion bodies. These are signs that the ability of that neuron to create and properly fold and move around proteins is impaired. What do proteins do for us? Fantastic, everything. So when you have a problem producing and moving around proteins, you have a problem with pretty much everything. And that's what we can see here. We shouldn't see protein aggregates. We'll also see the accumulation of neurofilaments. These are intermediate filaments in neurons. So these are parts of the cytoskeleton that help keep the cell together. When you have cytoskeletal issues, you will have difficulties moving proteins around, although they don't use intermediate filaments. Intermediate filaments still help stabilize the cell. And they can get in the way of microtubule transport. Hopefully we recall that the axon is unable to make proteins. All happens in the cell body, so you have to transport proteins down your axon. So when you see the accumulation of neurofilaments in the cell body or the neurites, that tells you you have cytoskeletal issues. You're probably going to have difficulties transporting things along your axon. We'll see. You'll see reactive gliosis and demyelination in motor pathways, not sensory pathways though. So here's the spinal cord of a person without ALS. Here's an ALS patient. The blue is showing us myelin. This is a myelin stain. We should see plenty of myelin, except for where we have neuron cell bodies. So this looks totally normal. This is great. Notice the difference here. See how the posterior columns are standing out? They're still well myelinated. What's not myelinated would be your anterior or your lateral corticospinal tracts. In other words, your motor pathways. We're seeing the loss of motor neurons. We're seeing the loss of myelin, of course. If you have no axon to myelinate, you're not going to have myelin. And we see reactive gliosis. This is nothing new. This is still going to be bad for neurons. It's still going to affect glutamate reuptake. We'll see that in a bit. The disease mechanism is multifold and not at all what you'd expect. When you read that SOD1 neutralizes free radicals, of course you're going to expect the accumulation of free radicals. Loss of function. That's just simply not the case. Down here, we're looking at the number of motor neurons. They have larger axons. And sensory neurons. Their axons are a little bit smaller. So we can distinguish between those two based on size. And it works pretty well. Here's your 1-year-old regular mouse, just so we know our baseline. Here is a mutant SOD1. So when they express mutant SOD1 here, loss of motor neurons, sensory neurons are pretty similar. Here's a haplozygous mouse, so only one copy of SOD1, not two. Should have two, only one though. We have lower levels of normal SOD1, and here's our mutant. Pretty similar. Here they've gotten rid of endogenous SOD1, so there's presumably much lower levels of SOD1 activity, but we're seeing the same degree of degeneration because of this mutant. If you didn't have this here, you'd see no degeneration. And then when they overexpress normal SOD1, this should rescue it. If it is indeed a loss of function, nope, no rescue, not a loss of function. It's a gain of function. And that function, unfortunately, is to cause the accumulation of neurofilaments and impair axonal transport. It's as bad as it sounds. Here are the data for those of you who care. We're looking at the movement of mitochondria, believe it or not. Time is in the vertical here. So we start at the top and we move down. If your white line stays vertical, it didn't move. See this one drifting to the right? That one moved in the anterograde fashion. These down here are only drifting to the left, which staying still. All right, so this is a normal mouse. Overexpress a whole bunch of SOD1. That's normal. Express that mutant SOD1. Here's the summary. Let's have a look. So the percentage of mitochondria that were moving around drops. So our organelles, our proteins, are less mobile. That sucks for the axon. It can't make proteins. Your proteins have to be mobile. If you can't move proteins down your axon, your presynaptic terminal depletes. No presynaptic terminal, no synapse. No synapse, same thing as no neuron. If we look at anterograde transport, we see that's where the big drop down goes. Far less anterograde transport with mutant SOD1. Retrograde seems fine for whatever reason. When you look across all of the ALS-associated mutations, they all kind of center around improper production of protein and shuttling of that protein within the neuron. So with SOD1, we're seeing this impaired axonal transport. In other cases, PT536 is Rna Binding Protein. It helps move RNA from the nucleus to the cytoplasm. If you don't do that, you have no mRNA to make protein with. That would be terrible. And then there's this C9 Open Reading Frame 72, and it's exactly what it sounds like, a gene. I don't know what it is. But the mutation in it should impair RNA metabolism, and that's going to be my extra point. So just tuck that in the back for right now. It's going to be just like what we talked about with Huntington's. The story that I want you to think about, just this. Across all your mutations, there's an inability to properly produce proteins and move them around the cell. Motor neurons have incredibly long axons. We're going from the cortex down to the brainstem or probably the spinal cord, and then from the spinal cord out to submuscle. That's quite the distance to traverse. So any impairment in axonal transport is going to be devastating to motor neurons. They don't just have local circuits. They span from head to toe. Yes?
When you say it's similar to Huntington's, is that like where it's searching for something that's not?
Precisely.
Okay.
Yes, and I will try to illustrate that far more clearly than just with this. Now, the disease mechanism could also have everything to do with glia. As a matter of fact, if you express mutant SOD1 in astrocytes only and not neurons, you'll see the death of motor neurons. This is what we call non-cell autonomous toxicity. It's a pretty cool idea. We'll go through it real quick. Here's our reactive gliosis. Here we're seeing the formation of a glial scar, believe it or not. Here's a regular mouse. Everything's fine, nothing to see here. Here is a mouse expressing mutant SOD1. You can see we're starting to have an increase in this glial fibrillary acidic protein. When you see an increase in this, that says glial scar. That's all we need to know. The darker it is, the bigger the glial scar. And at the end of the disease, check it out. A lot of reactive gliosis. Great. Who cares? Well, all right. Well, the nearby neurons care, it turns out. Astrocytes are not packing peanuts. They do things. And one of the very important things that they do is buffer glutamate. Do we recall from lecture 1, when you knock out glutamate transporters, you see seizure activity in mice and they die really quickly? If not, see lecture 1, it's in there. Here we're seeing the loss of glutamate transporter expression. That's what E2 is. Here's your normal mouse. Here's the spinal cord. Dorsal, ventral. Here's where our motor neurons live. The darker it is, the more glutamate transporter you have. Let's have a look in our ALS model. Uh-oh. Here in the ventral horn, no glutamate transporters. If you don't pick up glutamate, it builds up. You get glutamate excitotoxicity. This is a common disease mechanism. It's still just as true as it was when we talked about it in stroke or cancer. So it's still bad. You're still going to get an elevated level of glutamate, and we see this in ALS patients. They have higher circulating levels of glutamate in their CSF. So this is not just something that happens in mice. This happens in people too. That's why we care about it. And that's going to cause that excitotoxicity. Hyperactivation of NMDA receptors, just like we saw in Alzheimer's disease and stroke and cancer. And now ALS. It's back. This is still lethal. You got to keep glutamate levels low. You don't want it building up to toxic levels and hyperactivating your neurons and having them have an elevation in calcium. What's an elevation in intracellular calcium going to cause? Apoptosis. Fantastic. Hey, we've associated that together. Wonderful. Your NMDA receptors are working. Now, the progression is rapid compared to these other diseases where it might take decades to come on. Once you see symptoms, you likely only have a few years left. With bulbar onset, you have, 2 to 3, and with your more lateral onset, 3 to 5 years. Unless you're Stephen Hawking, then you have about 55 years for whatever reason he survived that long. The treatment is really just supportive. If they have respiratory issues, offer respiratory support. You guys will know what this means. I'm going to even attempt it, so you can read that point. And then, of course, speech devices. When you have difficulty speaking, but your eyes can still move just fine, you can just use eye tracking and you can communicate that way. All right. Any questions? Yes?
So are you saying that seizures are common in people with ALS? When you were talking about
No. I was just trying to point out that whenever your glutamate transporters aren't working, you see hyperexcitability of neurons, and that causes them to die. So your motor neurons get hyperexcited, and they die. That's the story here. So down there in the ventral horn, if you're not buffering that glutamate, it builds up to a toxic level, and your lower motor neurons die off. Yeah. Seizure activity is going to be driven through widespread activation of the cortex. This is more local in the spinal cord. Very good question. Yeah.
Okay, I'm still a little confused with like SOD1 mutations. So is it gain of function or is it loss of function?
Well, when we knock it out, we don't see the loss of motor neurons. So I would say it's not loss of function.
Okay.
And as a matter of fact, if you look at the enzymatic activity of SOD1 mutants, many of them still have upwards of 90% enzymatic activity. That's pretty close to 100. Far from 0. So all signs point to gain of function. And that gain of function would be abnormal axonal transport. Now, why do we need axonal transport? Proteins, exactly. You got to move proteins down your axon because you can't make proteins in the presynaptic terminals. They don't have the machinery to do it. They don't have ribosomes. What are they going to do? So you make it in the cell body and you ship it down. So these SOD1 mutants interfere with axonal transport. This leads to the starving of synapses, just like we saw in Alzheimer's disease. There's a lot of commonalities here. We'll have a lecture on that. And so when your synapses starve, your neurons are not long for this world. They require active synaptic communication to stay alive. Any other questions? Yeah.
We talked about,
Okay.
So impaired axonal transport being like one possible cause, and also reactive gliosis. So are you saying it's both, or we don't know which one?
Oh, I'd say it's likely a combination of many things going wrong, and that's why this disease progresses so rapidly. Yeah. So it doesn't have to be one or the other. Like the nature versus nurture argument. Why do we have to choose? Both. Isn't that the simpler answer? So it's a combination of all these things, I would imagine, playing a role. Is that a question, William? Okay. Well, I have some. Let's make sure we can answer these, and then I'll try to tell you something about genes first.
Alright, so
Okay.
So on the thing like
Yeah.
so
Well, in all likelihood, they're buffering reactive most mutations aren't effective. Instead, what they're going to do is cause SOD1 to do something. So that's gain of function. I think that is because of neuroenzymatic activity, the lack of utilizing and toxic and the inability to rescue your motor neuron loss by overexpressing wild type soluble of the system loss of function. If we express higher levels of the normal protein, everything is fine. That would work in the loss of function, but it doesn't. So rather than impaired free radicalism, instead, I would say that the cell-death soluble mutation has everything to do with impaired action transport of neurons and impaired glutamate uptake. Both of those are going to
It was just, I was just really confused when I was like looking over the quiz and trying to answer the questions because it says impaired radical scavenging, but then it's like, but then kind of not, and so I was like, oh,
Yeah.
okay, I don't know what the actual answer is here.
So you would expect it to do that. That's not what happened.
Okay.
Yeah. But it is very confusing. Yeah. And for the longest time, that's what we thought was going on. We
But
thought.
not for the reason that you thought. That's alright, you got it right. Different. Yeah.
Okay.
Okay. Yes, that makes, yeah. Okay. Correction. Well, that's what. That's what he was explaining. He was explaining why that would be false, but like the reasoning that we were talking about last night. It's so ridiculous. It's okay. Oh, here. Yeah. I'm sorry. It's upper motor.
All right, I hate to cut the discussion short. It is nice to hear people actually talking today. Let's go through these, just so we have some answer before we head out. Stephanie Smith.
Hi.
Clinical symptoms of ALS.
So the sensory function is still intact. You're pretty much saying like muscle weakness and muscle atrophy, and then depending which one of upper motor or lower motor, it's like the changes in terms of farther into it, in terms of tone and stuff.
Okay, so we're gonna have motor neuron loss, sensory neurons are just fine. Does this progress at all?
Mm-hmm.
Okay. Progressively? Or,
yes.
Okay, great.
Very
All right. Fantastic. And muscle wasting?
Yeah.
It's the amyotrophic. Okay, great. Great. So how can we tell the difference between upper and lower motor neuron weakness? Noah.
In upper motor neuron weakness, you have abnormal reflexes and like you have increased muscle spasticity. But for lower motor neurons, there's absence of reflex and then decreased tone, increased muscle wasting.
Okay, great. Does that all make sense? Does it make sense that you can't have a reflex if there's no motor neuron innervating that muscle? Okay, good. Good. So you should see a reduction in the amplitude of reflexes when you have lower motor neuron damage. But what about that upper motor neuron damage? Why would that lead to just hyperactive reflexes? Okay, great. Great. Part of what upper motor neurons do is inhibit reflexes. Yes. All right. Bulbar and pseudobulbar palsy. Monica.
Bulbar is death of lower motor neurons in the brainstem. Pseudobulbar is death of upper motor neurons in innervated.
Great. They're both going to affect the brainstem, but one's lower, one's upper. So you'll see differences in the reflexes for your cranial nerves as a result. Both have poor prognosis. Why is that? There we go. Brainstem does very important stuff for us. So if you have degeneration affecting the brainstem, you have degeneration affecting vital functions. Okay. Cytological hallmarks. In other words, what are the cellular changes that we see? Brianne.
So we see like muscle wasting, we see the inclusion bodies. In addition to we see like demyelination in like the spinal cord, but not in the posterior columns. And then we see like the reactive gliosis.
Great. And then neurofilaments kind of accumulating where they shouldn't,
Yes.
telling you your cytoskeleton is not working properly. Okay. Sod 1. It is a bit confusing. What's its normal function? Cody.
You get rid of free radicals, but in this case it's a gain of function, so you have neurofilament aggregation, which you can't make protein and you can't transfer protein, and so that's not good. And then you also get the gliosis, so astrocytes can't take up glutamate, and that leads to
Excitotoxicity, yeah, exactly. Excitation that's toxic. Very good. Okay, so we got our neuronal and glial impairments right there. Any questions on that? Ask Cody. All right, gene expression. We've all heard the central dogma. We go DNA, RNA, protein. This is true. I'm not going to tell you otherwise. RNA is not just a middleman, though. It does other things. So, transcription. Wow, I'm a moron. Should say transcription there. That's where we read our DNA and create RNA from it. So translate this to transcription, please. It's right up here. And then translation is where we take that RNA and turn it into a protein. But there is quite a bit going on between here and here. And that would be the processing of that RNA. We're not prokaryotes. I think I can safely say that. So we have to fix our RNA, just like I fixed that. Sometimes you need to do a little editing. We can do that. We can edit single nucleotides, or we can take out big chunks. One of the things that you're going to have to do is modify your ends. The 5' and the 3' ends are modified to say this RNA is ready to leave the nucleus. How else would your nuclear export proteins know? Which RNA should I take out? Which one's been edited? Well, just look for your 5' cap and this poly A tail. That's it. Look for those. That's going to say this is ready to leave. There are other things besides just adding a little nucleotide here and then somewhere between 50 and 250 nucleotides at the end. We also remove sections. Introns. Introns don't make it into the final product. They're removed. And you keep the exons. And now you're left with your ready to read messenger RNA. And you might be thinking, why would you do this? This is a waste of time. Why would you spend the energy to make these introns? Because we don't always use the same bits as introns. We can have alternative splicing. So you can have one gene make 1,000 different proteins just by selecting a different combination of exons. This is one of the ways that our cells are able to tell itself from its neighbor. How does a brain cell know as it's spreading out its dendrites, am I hitting my own or am I hitting a neighbor's dendrite? Through alternative splicing. So this is pretty nifty. There is a reason why we take all the time to put in that junk that we later take out. Editing is very important. It's true for your writing and it's true for your gene expression as well. Now, the RNA that we've looked at so far is all single-stranded. But not all RNA remains single-stranded. Some of it folds back on itself. to form a hairpin, and it looks like a little hairpin. That's why we call it that. There are complementary sequences in that RNA that allows it to fold back and base pair. And this certainly looks double-stranded to me. It also looks double-stranded to dicer. This is an enzyme that dices up double-stranded RNA, hence the name dicer. These little bits are the microRNAs that every cell makes to help fine-tune its gene expression. These little microRNAs get loaded into the RNA-induced silencing complex, or RISC, if you're a board game fan, and that looks for complementary sequences, identifies them, and then silences them. There's a couple ways of silencing. You can cut up the RNA, that's what we see here. Here's my RISC. I got my template. What's complementary to this? That's that double-stranded RNA I want to get rid of. Oh, there it is. I'm going to chop it up. You can also prevent association with the ribosome. Same function. No protein is getting made from this. They can also find that genetic sequence in the DNA and recruit a bunch of methyltransferases there to cause the DNA to compact up, so you can't read it. If you're going to read a book, you have to open it. If you're going to read your DNA, you have to open it. So by compacting the DNA, you can silence that gene in the genomic level. Double-stranded RNA turns off genes. Here's how it does it. There are many ways that it does it, but double-stranded RNA does that, and our cells do this all the time. MicroRNAs are what make neurons unique from each other. They're specific types of microRNAs that they produce, and there are hundreds of these, are going to fine-tune its gene expression. So it's very important that we're able to load up RISC with appropriate microRNAs and make sure we're expressing the right genes. When we have a problem with this, that's where we get toxicity. So I just want to present this data to you very quickly so we can see RNA-based toxicity, as the title says. Sometimes titles are informative. This is one of those cases. In their experiments, they decided they're going to look at polyglutamine in ataxin-3. This is another polyglutamine-associated gene. It causes spinocerebellar ataxia. That's why it's SCA. So it causes degeneration of Purkinje neurons to the cerebellum, and that leads to ataxia, gait abnormalities, and difficulties moving limbs. So they have a CAG expansion, just like in Huntington's, but it's a different gene, so it affects different neurons, because different neurons express different genes. Both of these sequences here are going to give you 78 glutamines. One's going to use only CAG, just like we see in real life. The other one's going to use CAA, that encodes glutamine, and CAG, that also encodes glutamine. They're different RNA. Same protein, different RNA. They do this in flies because they're cheap, they're quick, and you can easily see neurodegeneration by just simply looking at the eye. There's a whole bunch of nervous tissue in the eye. So as you see neurodegeneration, you'll see retinal degeneration in the fly. They have compound eyes made up of individual subunits called ommatidia. Fun to say. Within each ommatidium, you have 7 different photoreceptors called rhabdomeres. So 7 would be normal. Anything less than 7 would tell you degeneration. You can just see it visually. Here's your wild-type fly. You can count in there. In each of those 7 ommatidia, there's 7 rhabdomeres. Oh, not here, not here, not here. We're seeing degeneration whenever we have that CAG repeat. Over here, you can see most of them are still 7. Same polyglutamine, different RNA, CAA-CAG. Now that's not to say that nothing is going on here. We still see reduced survivability here. So green is just a wild type set of flies. Some of them died, not that many. When you express CAG repeats, marked toxicity. Half of them are dead after about 2 weeks. It takes closer to 4 weeks, maybe 3.5 weeks, to see half of them dead when you have CAA, CAG. Still polyglutamine, same protein, different RNA. This is where we can definitively say it's RNA-based toxicity. Here, they put the polyglutamine, well, not polyglutamine, the poly-CAG repeat in the untranslated region of the RNA. Not every bit of RNA gets turned into protein. This is the coding sequence for a red fluorescent protein. So it'll just tell you where this is being expressed, because it's red. Here's where it's expressed. In the untranslated region, no protein is going to get made. And when you look at the size of the protein, depending if you have 0, 100, or 250 CAG repeats, the protein doesn't change size. It's the exact same size. If it were bigger, it would move upward, but it doesn't. Purely in the RNA, no protein at all. Look at these big holes that develop in the retina and the brain of this little fruit fly here. Has nothing to do with protein. Everything to do with RNA. Now, why is that the case? Well, let's look up what CAG repeated 40 times would look like. That looks a hell of a lot like a hairpin to me. How about you? I see hairpin here. I see hairpin here too. That should be GGGCC. That's the expansion we see in ALS. Ah, they look very similar. Interesting. Almost like I have a point here. And then over here, CAACAG. Not a hairpin. Bubbles. That's not double-stranded RNA. That's just bubbly RNA. Not the same thing. That's not going to get recognized by Dicer, loaded into RISC, and then used to silence some gene. This is always going on in our neurons. This is what helps them express the correct genes. If you futz with microRNAs, you will kill that neuron. There is no doubt about it. And these CAG repeats that we see in Huntington's, or spinal cerebellar ataxia, or the GGGGCC repeats, also a ton of fun to say, that we see in ALS. These are going to create hairpins, pseudo double-stranded RNA that gets loaded into RISC. And when it goes looking to see what should I turn off, it's looking for something that doesn't exist. Now you've taken your RISC, which should be used to use an actual microRNA, something that you really do want to target and turn off. It can't get loaded anymore. It's loaded up with this junk. So you're taking parts of your microRNA machinery and giving them something to do that they'll never get done. And so the genes that should have been silenced don't get silenced. When you mess with gene expression, you kill a neuron. Very easy to do. This is how it happens. Huntington's, now in ALS. Hopefully that makes a bit more sense. If you make something that looks like double-stranded RNA, you're going to mess with microRNA processing. You're going to change gene expression in that neuron, and it's going to die. Okay, that's all I got for you. I do thank you for the extra 3.5 minutes. If you have any questions, let me know. I'm here. If not, I'll see you Wednesday. Tomorrow, too.
I've checked every day.